And our next two stories are going to be stuff that's very, very small. There's been all this news or rumors and gossip that there's been turmoil on the set of the new Avengers movie.
Yeah, I saw a lot of memes about it. Yeah.
And the details are a little fuzzy. Mm-hmm. But apparently, Robert Downey Jr., RDJ, Iron Man, who is the tentpole of the whole franchise, the only reason it exists, has been having some conflicts with one of the more aggressive characters in the Marvel Universe, Deadpool, a.k.a. Ryan Reynolds.
Right.
Who apparently can't stop fighting each other on set. But weirdly, how this connects to our story is our story number 2 is that apparently a virus and bacteria are teaming up and are able to battle cancer as if it's Thanos in Endgame.
Yeah.
There are no 5 rings on the thingy.
No.
But
this team up could actually change the game for cancer therapy.
So this story is coming out of Columbia Engineering. Researchers at Columbia Engineering. Byline is researchers at Columbia Engineering have built a cancer therapy that makes bacteria and viruses work as a team. It was published in Nature Biomedical Engineering. And the Synthetic Biological Systems Lab shows how their system hides a virus inside a tumor-seeking bacterium and smuggles it past the immune system and unleashes in it inside these cancerous tumors, unleashes the force.
It's insane.
So there's an immigration story here.
Yeah.
There's a smuggling story here. Columbia was obviously the center point for a lot of controversy as it relates to
No, they're doing good work.
Science and research and funding. But tell me, this sounds like a great buddy cop movie.
It is. It is exactly that.
Okay.
Yeah, it is exactly that. It's like two cops that are teaming up.
Yes.
To take care of a bad guy. It's almost like what's the one movie where they take the criminals and then they're like Suicide Squad.
Oh yeah.
Right? Where they're like, they take the criminals and then they're like, okay, we're going to make you good now. And then you're going to get the even worse bad guys. That's what this is.
Okay. Got it.
Right? You know what I mean?
Yeah.
Because it's like bacteria. We don't like that.
Right.
Viruses. We don't like that. But the two of them together are now fighting cancer, which we really don't like.
It's a super evil duo to fight the even more
evil duo.
doer.
Yeah. It's that the meme with Thor being like, I know I can't, but she can. You know what I mean? But it's like this virus bacteria chimera that they've created. It's an incredible paradigm shift in cancer treatment.
So what I understand about cancer treatment, which is very little, is fundamentally it's very like the existing options we have for the patients, whether it's chemo, all this, it's like very hard on the body.
Yeah, it's very hard on the body, and it's also very non-specific. That's the main thing.
It's a shotgun approach.
Yeah. It's just like with chemo, all you're doing is targeting cells that replicate really fast. Okay, so you're getting rid of the cancer cells, but you're also getting rid of hair cells because hair follicles because they replicate really fast. Bone marrow, they replicate really fast. Digestive cells that replicate really fast. That's why you get nausea, anemia, hair loss.
Yes.
So it's non-specific.
Yes.
And then with radiotherapy, it's like sort of like, you're just sort of blasting some location. And sure, you can get specific with like proton therapy and things like that. But at the end of the day, it's not like targeting from a biological sense only the cancer cells, right? So over the years, we've had these things called adaptive therapies. Okay. And adaptive therapies, what they've done is sort of target only the cancer cells and leave everything else alone.
Right.
Okay. There's been sort of a bunch of approaches. There's been this thing called CAR T therapy, which is where you take the body's own immune cells and then you engineer those immune cells to then target the antigen, which is like sort of a marker on these cancer cells. So you train the body's own immune system to then target these cancer cells and recognize these cancer cells. The problem with that therapy is, okay, it's got to be extremely specific to the patient. You got to literally extract immune cells from the patient, and then you've got to engineer those
On a per patient basis.
On a per patient basis. And then also at the end of the day, a lot of these cancers are clever, dude. They've created methods to actually create cold tumors. It's what we call when they suppress the immune response. Okay? Even with the CAR T and all this stuff, like they're suppressing the immune response actively because they're secreting stuff that confuses the immune system.
It's like you say, oh, we're gonna build a wall, and they're like, we have ladders.
Yeah, and it's like, or like we're making the wall invisible, and then it's like, oh, okay, now I can't, my immune system can't actually see everything, right?
Right.
So, over the years, we've shifted towards something called adaptive therapies, which are these like single agent biotherapies where we're now actually leveraging biology itself to attack biology. Okay. So there's two approaches. One approach is to use bacteria. Okay. What we do is we take some bacteria and we try to get that bacteria to attack cancer.
Yes.
Okay. There's some advantages to this. The advantage is there's certain types of bacteria like Salmonella that like anoxic environments. So environments that don't have a lot of oxygen. Tumors tend to have not a lot of oxygen, especially at their core, because the blood vessels are on the periphery, and at the core, the oxygen isn't getting to it. So the bacteria naturally are like, ooh, I like that.
Yeah.
And then they get to the core, and then they start sort of, destroying the cancer from within. The problem with that is they get to the core, but they're not really attacking the periphery, and they're not attacking like maybe a metastasis that happened somewhere else, right? And the other problem obviously is like, okay, like you're literally giving me Salmonella, right? You can genetically engineer and everything, but like,
it's still bacteria.
You're giving me a pathogenic bacteria, right? And there's a risk of systemic infection and dose-related toxicity and all this other kind of stuff. Okay. The other way to approach this problem is with viruses. Okay. You have something called Oncolytic virotherapy, where you take viruses that attack cancer cells and you inject that into the patient, right? Now, with oncolytic virotherapy, there's two problems. One is the delivery challenge. Okay, you got to really get into the tumor and like put that in there.
Inject direct.
Okay, inject direct. So if there's a tumor that's very, very deep, or if there's like some random tumors elsewhere that haven't been identified, out of luck.
It's not like a systemic approach.
Yeah,
it's localized.
and the other thing is, the immune system of the body has already been primed with a bunch of viruses that you've seen over the years, right? So the immune system itself is going to be like, you're not getting in. What are you doing? And then the immune system is going to attack that virus, right?
Makes sense.
So this particular therapy called the CAPSID, CAPPSID, right, with two P's.
Yes.
This particular therapy is getting the best of both worlds. Okay?
Yes.
It's combining the virus, which is really good at killing the cancer cells.
Yes.
But it's hard getting in. It's got a tough time getting in.
It can't get past the security at the door.
So it's the virus is becoming like the Greek soldiers inside the Trojan horse.
Yeah.
And the bacteria becomes a Trojan horse. Okay, because the bacteria has an easy time getting into the center of the tumor, right? And it knows where to go because of the anoxic environment. It's seeking this place without any oxygen, so it goes to the center of the tumor. So what this team at Columbia Engineering is doing is combining both of those therapies to give this like two-pronged approach. It's a synergistic biotherapy that combines the best of both worlds to target the tumor and then infect the tumor with the viruses and then also have that viral infection spread to other tumors in the body. Dude, it's like it's almost out of science fiction. I can't, like, the amount of stuff that needed to happen for this to be a reality, right? Where we're now manipulating viruses, bacteria together
in combination,
where they're both burning together. Yeah, dude, it's actually insane.
It looks like this. So CAPSID, it looks like it's short for coordinated activity of prokaryote and picornavirus. of safe intercellular delivery.
Yes, prokaryote means a type of life that doesn't have a nucleus, so that's bacteria.
Right.
Okay, and coronavirus is the particular type of virus that they've used to genetically engineer to create this kind of viral therapy.
Not coronavirus.
No, not coronavirus. Yeah, this is like a type of, it's a Seneca virus.
Yes,
Seneca virus A.
Yes.
Yeah.
Yes.
Yeah.
It's
dude, it's a game changer, and there's a lot of like hurdles that you have to get through. Right. Because like, okay, for example, one thing would be like, okay, I've got this bacteria and it's got a viral DNA inside. Like, what's to guarantee that the viral DNA isn't just going to like express itself not in the tumor? What I'd like is for the virus to only target the tumor, right? I don't want it to, I got a bunch of healthy cells, right? I don't want a viral infection everywhere.
Then we're back to the same issue with the chemo and radiotherapy, which is it's non-specific.
Which non-specific. So how do I make this specific, right? So they engineered a way such that the viral DNA would only be expressed once the bacteria was inside the tumor.
That's sick. Once it's in the club, then
then that's when it does all of its shit. Right? And the way they did that was they attached it to a promoter, which we talked about. Right? Like the gene expression that's happening would only be expressed once the bacteria was in its environment where it wants to be pathogenic. Okay? And then once it did that, the viral DNA now starts transcribing. The other thing that was really cool that they did was they included a T7 RNA polymerase. So you've got this DNA that is really the viral DNA inside the bacteria, right?
Yep.
And what you want to do, usually what you want to do is viruses, they hijack the host cell, right? And then they replicate DNA using the host cell.
Yes.
This thing's got its own machinery to replicate the viral DNA. So it doesn't need to require the host cell, because the host cell is cancerous. It could be doing all sorts of random crap. Right? Like it's like replicating like crazy.
Yes.
So it's doing all sorts of crap.
And so it's maybe not as dependable as
exactly. We don't want to depend on this dysfunctional machinery of cancer cells in order to replicate the virus that is going to destroy the cancer cell.
Yep.
Right? So we've created our own little polymerase that will do that. Which is really cool.
Yes.
Yeah.
Yes. I want to make a quick shout out to Charles M. Rice, who is an expert in virology at the Rockefeller University, who was a part of the study and collaborated with the Columbia team. So I want to make sure we give...
Right across the island of Manhattan.
Oh, there
Rockefeller and Columbia. They're east side versus west side.
A little quick yacht trip across there. So just making sure that we give Charles a shout out there, as well as, again, the team over at Columbia.
Yeah, dude, it was insane. And, dude... You want to hear the experiment that was like nail in the coffin?
Because we love talking about experimental design.
The experimental design here is amazing. So they got these mice models.
Yes.
Again, transgenic mice models that sort of mimic the small cell lung cancer. Okay, 100% survival rate.
Jesus.
Okay. Here's what they did. And not just that. Okay, here's what's crazy. Okay, so this mouse model has a tumor in each of its lungs. Okay, right flank, left flank. What they did was they injected this therapy into the right flank, and the therapy killed the cancer in the right flank, and then it went to the left flank and killed it there too. Right, because the virus inside the right flank lung does the infection, creates these antibodies, right? And these viral particles. Those viral particles then travel through the bloodstream and start targeting the cancer in the other location as well, right? And the whole point was now the immune system is now overrun, because before, if we just inject the viral particles, the immune system is going to be like, that's not enough. But now we have this full-fledged viral infection. But all this virus is targeting are cancer cells. Right? It just wants to eat cancer cells because that's what the particles are sort of lock and keying into.
Yes.
You know?
Yeah.
So now you can put this into one part, and as long as if it's the same cancer that's spread to other parts of the body, it'll target those other cancers.
This is so important.
It's so cool.
This is so important because this goes back to what we talked about at the beginning of the story, which is it's no longer non-specific. And not only is it non-specific, you can insert it into the sort of body in a singular location, and it will independently locate and then also kill other similar cancer locations for that exact same targeted focus. Which, again, for patients, like this comes back to like, what is the impact for, research like this on real people? It's like these therapies can become can be more effective with less of an impact on the body.
Exactly.
In these like other ways in which the non-specific therapies, which we have available to us now.
Yeah, like CAR T therapy is a specific therapy that happens like patient by patient, but it's got problems, right? Like the CAR T therapy, these immune cells, for example, they can't get to the center of the tumor,
right?
Because the tumor has these natural mechanisms where it wants to fight off the immune cells,
right?
Right. On the other and then also it's CAR T therapy. You got to train these immune cells to identify specific antigens. But cancer is just like such a to work with because cancer is always mutating. Right. So, okay, you don't like this antigen, I'll give you another one. And then the CAR T therapy is not going to work because it's only trained on a single antigen. Right. And now I've just changed the shape to how you recognize me. And like my ID is now different.
Right.
And so you'll just let me go.
Access denied.
And so here with the viral DNA is constantly evolving as well. Right. So it can mitigate this heterogeneity, this like difference that's happening with these rapidly mutating cancer lines, right?
Which is one of the key aspects of why cancer is such a pernicious issue to
deal with. It's so hard because it changes so fast. But this, we're like harnessing the fact that viruses also
do that,
and they create like all of these different versions,
and then when one works,
and then that'll go, and then like it'll find another version that'll work. It's like synthetic biology, which is crazy to think about. Like we're literally engineering like little tiny micro molecular robots to now just target these bad guys, these cancer cells, right? We're far away from like FDA approval,
100%.
Right? There's lots of concerns. Obviously, like I talked about mutation just now, right? Like what's stopping this virus from mutating into like its original version of where like I hate it. Right. So it's like we've created our own micromolecular suicide squad. But we haven't, like, there's still the concerns, but they are still the suicide squad. There's still the suicide squad. So like in order to get to actually like human trials, because right now they've done it on mice and they've done it on in vitro sort of human cell lines and things like that. In order to get it to sort of FDA approval, we have to first of all mitigate the risks. One of the ways that they're actually doing it, and they described it in this paper, is instead of a single nucleotide mutation that will take the virus and put it back into its wild type, which is the part that we don't like, they made it 2 mutations. Okay, so what that means is now, like, if the probability of like going to a mutation was 1/100, now it's geometric. So it's 1/100 squared, so 1/10,000, which is like obviously way better than 1/100. You can imagine like doing this for multiple single nucleotide mutations. We also need to test it on higher order organisms, right? Right now we did it on mice. We need to like start graduating and slowly going towards human beings. But I think it's extremely promising. The other thing is like this thing, like it's currently been targeted for neuroendocrine like cancer cells. So it's like small cell lung cancer and things like that. But one can imagine this is a modular thing, right? The bacteria could have any sort of virus. And the bacteria could be any other kind of bacteria that we sort of engineered to get in here. So it's this like, Lego block thing that we talk about all the time, where it's like we can have different types of Lego blocks that we put in here and here. It's this modular thing that we can adapt to different kinds of viruses. I think it's extremely special.
Adaptive biotherapy. It was what you sort of started with. I mean, there's a key insight here, fundamentally. That has so many different paths that it can now go down because it's a key. It's similar to how CRISPR was a key insight.
That's right.
That had then multiple of these like
because it was modular at the end of the day.
At the end of the day, it's not just an acute, specific, singular insight. And like that's what's again so fascinating about this study in particular, in terms of now instead of there being 1 door, 2 doors, 3 doors.
Now it's like we can just like mix and match and get a bunch of doors. Every door you want.
is potentially available.
Yeah, it's pretty
obviously
cool.
with the caveats of downstream research to
Yeah,
mitigate.
and obviously we need to make sure it's safe, but I think this is really promising.
Fascinating. We're moving into a place again. We talk about this a lot between the fundamental research stuff in sort of this biotech arena, and then you add the ability to then sort of rapidly iterate with things like AI.
Yep.
Progress is going to continue.
Yeah,
to be
it's gonna be really cool. Yeah,
fascinating.
It's gonna be awesome.